Hello everyone, welcome to Biotech Notebook. In this video we are going to see in detail about the cells which all arises from this myeloid progenitors. So in the last video we have seen in detail about what is stem cell and what is hematopoietic stem cell and the cells which all arises from this lymphoid progenitor. That is, we have seen in detail about T helper cell, cytotoxic T cell, B cell and natural killer cells. Okay, so dendritic cell we will see in this video because this dendritic cell arises from both lymphoid progenitor and myeloid progenitor. So I covered this dendritic cell in this video. The cells which arises from myeloid progenitor are granulocyte, monocyte progenitor, dendritic cells, eosinophil progenitor, basophil progenitor, mast cells, erythroid progenitor and megakaryocytes. This granulocyte monocyte progenitor differentiates into monocyte and neutrophil. This monocyte circulates in blood and it differentiated into macrophage and dendritic cells in the tissues. Eosinophil progenitor differentiates into eosinophil, then basophil progenitor differentiates into basophil. This basophil circulates in blood and in tissues it differentiates into mast cell. Then erythroid progenitor differentiates into erythrocyte and megakaryocytes differentiated into platelets. Erythrocyte and megakaryocytes are not involved in our immune system. So except these two cells, we will see in detail about all these cells. Okay, so the cells that arises from myeloid lineages are red blood cells, then various types of white blood cells, that is granulocytes, monocytes, macrophage and dendritic cells. Okay, so these granulocytes also classified into neutrophils, basophils, eosinophils and mast cells. Okay, so let's start with granulocytes. So granulocytes are at the front lines of attack during an immune system. Okay, so these are the cells which first respond to your infections. Then these are considered as part of the innate immune system. So innate immune system means it is non-specific to the pathogens. Okay, it won't act very specifically to the pathogen. As granulocytes gives non-specific immune response, it considered as part of the innate immune system. Okay, then on the basis of difference in cellular morphology and the staining characteristics of cytoplasmic granules, we can classify these granulocytes into four types, that is neutrophils, basophils, eosinophils and mast cells. So next let's see the structural features of granulocytes. So common features of granulocytes. We are classifying granulocytes into four types: neutrophils, basophils, eosinophils and mast cells. All these cells have a general structural features. First let's see about this. Okay, so all these cells have multi-lobed nucleus. So these cells are known as polymorphonuclear leukocytes. Okay, polymorphonuclear leukocytes. So this multi-lobed nucleus make them visually distinctive and easily distinguishable from lymphocytes. Okay, lymphocytes doesn't have any multi-lobed nucleus, so whose nucleus are round in shape. Okay, lymphocytes nuclei are round in shape, but whereas leukocytes nuclei are multi-lobed. So these leukocytes are also known as polymorphonuclear leukocytes. Okay, the next structural feature of these leukocytes is cytoplasmic granules. Okay, the cytoplasm of all granulocytes is replete with granules. It has more granules in its cytoplasm. That's why these cells are known as granulocytes. Okay, these granules are released in response to contact with pathogens. Okay, so the granules present in the cytoplasm of granulocytes are released in response to contact with pathogens. Okay, these granules contains a variety of proteins with distinct function. So some proteins damage pathogens directly and some regulate trafficking and activity of other white bloods including lymphocytes. Okay, it will activate lymphocytes and it will attract lymphocytes to come to the infection place. Okay, then some contribute to the remodeling of tissue at the site of infection. So multilobed nucleus and cytoplasmic granules are the general structural features of granulocyte. Next, let's see in detail about each type of granulocytes. So the first type is neutrophils. Okay, neutrophils constitute the majority of circulating leukocytes. So 50% to 70% of your leukocytes are neutrophils. Okay, these are much more numerous than Eosinophils, basophils and mast cells. Okay, eosinophils are 1% to 3% and basophils are less than 1%. Mast cells also less than 1%. So neutrophils are more in number. It is 50% to 70%. Next, let's see the lifespan of neutrophils. After differentiation in the bone marrow, these neutrophils are released in peripheral blood and it circulate in the blood for 7 to 10 hours. Okay, after 7 to 10 hours, they migrate into tissues where they have a lifespan of only few days. So next, let's see functions of neutrophils. These neutrophils are the dominant first responders to infection. Then the main component of your pus or neutrophils, where they accumulate at the end of their short lives. Then four main antimicrobial functions of neutrophils are phagocytosis, degranulation, then release of nuclear material in the form of neutrophil extracellular traps, that is known as nets, then cytokine production. Next, let's see each functions of neutrophils. First, start with phagocytosis. So, phagocytosis not only takes place in neutrophils, in macrophages, neutrophils and dendritic cells in tissues and monocytes in the blood or the main cell types that carry out phagocytosis. Okay, then phagocytosis is nothing but the cellular uptake, that is eating of particulate material such as bacteria to eliminate them. Okay, so to eliminate a pathogen, these innate cells uses phagocytosis process. In this process, the first step is bacterium becomes attached to a membrane evagination, which is called pseudopodia. Okay, then bacterium is ingested into the cell and it forms phagosome. Okay, phagosome then fuses with the lysosome and it forms phagolysosome, and bacterium is killed and then digested by the lysosomal enzymes. Okay, so these lysosomal enzymes are present in the lysosome which kill the bacteria and it digests the bacteria. Okay, then the digested products are released from the cell. Okay, so this is the general process of phagocytosis. This process is taking place in neutrophil, macrophage, dendritic cells in tissues, and it also takes place in monocytes in the blood. Okay, then the next process is degranulation process. So degranulation process is the process in which neutrophils mobilizes its granules is known as degranulation. So as we know that neutrophils contains more number of granules. Okay, so it mobilizes its granules to some place is known as degranulation. Okay, so this process takes place at plasma membrane, and if it releases its granules into the extracellular space to kill the bacterium, then it is known as extracellular killing, or it will mobilize these granules to phagosome. Okay, in the process of phagocytosis, then it will kill the bacterium which is present inside the phagosome, then it is known as intracellular killing. As we know that neutrophils has more granules in its cytoplasm, so in these granules it has antimicrobial proteins, has defensins lysozyme, and also it has tissue remodeling proteins like elastase, collagenase, and also it has protease inhibitors. Okay, the next process is release of nuclear material in the form of neutrophil extracellular traps. Okay, that is known as nets. That means neutrophil extracellular traps. In this process, activated neutrophils releases its intracellular content into the extracellular space and forms a spider's web-like structure that can enmesh microbes and kill them. It surrounds the bacteria, it forms mesh-like structure and it will kill the pathogen. These nets appear to be largely composed of neutrophil DNA with associated histone along with high concentration of neutrophil granule which contains protease such as cathepsin G, elastase and proteinase 3. Okay, the last one is cytokine production. Okay, the cytokine is produced by neutrophils and also some other innate cells. Okay, so this cytokines are chemical signals. So neutrophils are recruited to the site of infection in response to this inflammatory molecule. Example, chemokines. Chemokine is a type of cytokine which is generated by innate cells and including other neutrophils. Okay, that have engaged a pathogen. So once a neutrophil or some other cells engage a pathogen, it will release these type of signal molecules, that is chemokines. Okay, then neutrophils are recruited to the site of infection in response to this inflammatory molecule chemokines. The next one is basophils. Basophils are non-phagocytic granulocytes. Basophils won't kill the pathogen by the process of phagocytosis. Okay, so non-phagocytic granulocytes, then it contains large granules filled with basophilic proteins. Functions of basophils or degranulation or extracellular killing. Okay, so phagocytosis process is also known as intracellular killing because it engulfs the bacteria or your pathogen and it will kill inside it. Okay, so degranulation means release of granules from the cell, from the cytoplasm. Okay, so this degranulation process is also known as extracellular killing, and phagocytosis is also known as intracellular killing. So in response to the binding of circulating antibodies, basophils release the contents of their granules. Okay, when these basophils release their granules in response to the binding of circulating antibodies. Okay. The next function is histamine production. Histamine is one of the best known proteins in basophilic granules. Okay, so this histamine increases blood vessels permeability and it smooths muscle activity. Okay, it enlarges as the blood vessel. Okay, thereby there will be a increased flow of your blood, then there will be a smooth muscle activity. And then next one is parasite attack. Okay, basophils are important in the immune response to parasites. Okay, particularly helminths infection such as those caused by roundworms, whipworms and hookworms. Okay, so basophils give immediate response to the helminths infection. These infections are caused by roundworms, whipworms and hookworms. Then like neutrophils, basophils also secrete cytokines that modulate the adaptive immune response. That means it will give signal to the T lymphocytes and B lymphocytes. Thereby it activates the adaptive immune response. Then next one is mast cells. Mast cells are released from the bone marrow into the blood as undifferentiated cells. Okay, these mast cells mature only after they leave the blood. Okay, in the blood these mast cells are undifferentiated cells. Okay, once they leave the blood, enter into the tissues, they get differentiated. Then these mast cells are found in wide variety of tissues including skin, connective tissues of various organs, then mucosal epithelial tissues of respiratory tract, genitourinary tract and digestive tracts. Then functions of mast cells are like circulating basophils. These mast cells have large number of cytoplasmic granules. These granules contains histamine, then pharmacologically active substances. Okay, then it plays an important role in the development of allergies. One is eosinophils. Like neutrophils, these eosinophils are motile phagocytic cells and it can migrate from the blood into the tissue spaces. Next is functions of these eosinophils. So first one is degranulation. So by degranulation, it attacks parasites, defends against multicellular parasite organism including worms. So these eosinophils clustering around invading worms, then it damages membranes of the worms by the activity of proteins released from eosinophils granules. Okay, that means the proteins released by eosinophil granules damages the worm's membrane. Okay, the next function is cytokine production. It secretes cytokines that regulate B and T lymphocytes. Thereby, it also influences the adaptive immune response. Next one is myeloid antigen presenting cells. Okay, so myeloid progenitors gives rise to a group of phagocytic cell, that is monocytes, macrophage and dendritic cells. These cells have professional antigen presenting cell function. Okay, in lymphocytes, B cells also known as antigen presenting cells. Okay, so myeloid antigen presenting cells are monocyte, macrophage, dendritic cells. Then lymphoid antigen presenting cell is B lymphocyte. Okay, so in the last video we have seen in detail about this antigen presentation by B cell to the T cell. Okay, let's see the functions of antigen presenting cells. Functions of antigen presenting cells. So these antigen presenting cells has the functions of phagocytosis, antigen presentation, and it also secretes some cytokines and co-stimulator molecules. Okay, so these myeloid antigen presenting cells are considered cellular bridges between the innate and adaptive immune system. As we know that innate immune systems are non-specific defense system and adaptive immune systems are specific immune defense system. So here these antigen presenting cells make a bridge between the innate and adaptive immune system, okay, by the two process. One is phagocytosis and then antigen presentation. So through phagocytosis process, non-specifically, these antigen presenting cells can ingest pathogen. It is ingest pathogen means it engulf the pathogen and it will digest all pathogenic proteins into peptides. Then it present these peptide antigens on their membrane surface to the T lymphocytes. Okay, so the present of these antigen to T lymphocytes is also known as antigen presentation. Okay, so the antigen presenting cells make contact with the pathogen at the site of infection and communicate this encounter to T lymphocytes in the lymph node. Thereby, it initiates the adaptive immune system. Then these antigen presenting cells can respond to pathogens and secrete some proteins, cytokines and co-stimulatory molecules that attract and activate other immune cells. That is, signal molecules attract other cells to the infected area. Okay, now let's see in detail about each type of antigen presenting cells, that is monocytes, macrophage and dendritic cells. So first let's see about monocytes. So monocytes are 5 to 10% of your white blood cells. Then first let's see the lifespan of monocytes. So during hematopoiesis in the bone marrow, these granulocyte monocyte progenitor cells differentiated into promonocyte in the bone marrow, then which leave the bone marrow and enter into the blood where they differentiate into monocytes. In blood, they further differentiated into mature monocytes. There are two broad categories of monocytes. One is inflammatory monocytes and another one is patrolling monocytes. Okay, these inflammatory monocytes enter tissues quickly in response to infection, but patrolling monocytes are a small group of cells that crawl slowly along blood vessels. Okay, so it provides a reservoir for the tissue resident monocytes in the absence of infection. So these patrolling monocytes provides a reservoir for tissue resident monocyte in the absence of infection. Okay, it resides in the tissue in the absence of infection. Next, let's see about macrophages. So monocytes migrate into tissues in response to infection and then differentiates into tissue specific macrophages. These tissue specific macrophages have different names based upon the tissue. These macrophages are termed as osteoclast in kidneys, mesangial cells, in brain, microglial cells, and in lungs, alveolar cells. Then in liver, it is known as Kupffer cells. Then functions of macrophages, it exhibit greater phagocytic activity and it increased ability to kill ingested microbes. Macrophage and Mast cells, both of these cell types have an important role in sensing infection, and it amplifies the immune responses through the production of cytokines, chemokines, soluble mediators, and it also facilitates the migration of other immune cells to the infection place by releasing these chemical signals. Dendritic cells acquire their name because they are covered with long membranous extensions that resembles the dendrites of nerve cells. Okay, that's why these cells are known as dendritic cells. Okay, so it resembles the dendrites of nerve cells. Okay, these extensions enable them to maximize the contact with other surroundings. Okay, there will be an increase in surface area. So it maximizes the contact with the surroundings. Then there are more diverse population of cells which arise from both the myeloid and lymphoid lineage of hematopoietic cells. Okay, so from lymphoid progenitor, dendritic cells are arised, and also from myeloid progenitor, your dendritic cells are arise. Okay, this is the structure of your dendritic cell. So this long membranous extensions or resembles the dendrites of nerve cells. That's why these termed as dendritic cell, and these extensions enable more surface area to contact with their surroundings. The next functions of dendritic cells. There are two types of functions for a dendritic cell. In one location it will capture the antigen, and in another location it will present the antigen. Okay, so outside lymph nodes, these immature form of dendritic cells monitor the body for signs of invasion by pathogens, and it captures the intruding foreign antigens. Okay, so outside the lymph nodes it will watch for antigens and it capture the antigen once it find. Okay, then they process these antigens and then it migrate to lymph node. Okay, in lymph node it will present the processed antigen to the naive T cells and it initiates the adaptive immune response. Okay, so in lymph nodes they present the antigen to naive T cells and initiates the adaptive immune response. Then this antigen capture outside the lymph node takes place in three ways. Okay, so dendritic cells may engulf antigen by phagocytosis, or it internalize the antigen by receptor mediated endocytosis, or it imbibe antigen by pinocytosis. Okay, so through a process of maturation they shift from antigen capturing phenotype to presentation of antigens to T cells. Okay, thank you for watching my videos. If you like my videos, please do like, share and subscribe. Thank you.